Hey there, STEAM team. This is Miss Danielle from the Kimball Library, bringing you another science experiment that you can do at home. Today, we're going to learn how to extract DNA. But first, we're going to have to know what DNA really is. What are we looking for? What are we doing? So, DNA stands for deoxyribonucleic acid. And that means it is the blueprint or the genetic code for all living things. Basically, the DNA is what tells a living thing what it's going to look like, what it's going to turn into, how it's going to grow, how it's going to live, etc., etc., etc. It's very, very important. And to that end, all of the little building blocks that make up anything alive, they're called cells. They each have their own little DNA that they keep tucked away in a special compartment. That special compartment of the cell is called the nucleus. I just threw a lot at you. Let's put it all together. So, all living things are made up of cells. Every cell has a nucleus. In the nucleus of the cell lives all the DNA. You guys with me so far? Of course you are. You're all brilliant. I know that. So let's look at our ingredients for our experiment today, and then I'll tell you guys what you're going to be doing. First we need dish soap, our trusty salt from last time, a bit of isopropyl alcohol or rubbing alcohol. While you're getting all your other ingredients ready, grab your rubbing alcohol and pop it in the freezer. We're going to come back for it later. Water and a strainer. You can also use a coffee filter, that will probably work. Anything that has sort of a fine mesh situation. The last thing you're going to need is, of course, something to take DNA out of. Now, typically, for this experiment, strawberries are used, specifically because they have so much DNA. They're just packed with it. So, if you have strawberries, that would be a really great choice. I will be using a cherry tomato because I found it in my kitchen. You can use pretty much any fruit or vegetable that you have. It should work just fine. Now, you're going to want to make sure that it is something that you can mash up easily. So, take your subject, as it were, and put it in a plastic bag and mash it up really well. The idea here is that we are smushing our subject and dividing up the cells that hold it together. Now, cells are too small still to be seen by the human eye, but if we separate the cells even a little bit, that will help us get more easily to the nucleus of each one. Now, we have our subject all mashed up and gross looking in our plastic bag here. The next thing we need to do is make our extraction solution. Now, let's see what we need for that. First of all, our water. Then our soap and our salt. When we mix these together and put them in with our mashed up subject, what they're going to do is dissolve the barrier around the cell. So each cell, we already know, has a nucleus. What it also has, and what we want to get at, is what's called the cell membrane. And that is basically like a barrier or a wall around the cell that keeps it all intact, keeps it cozy, keeps it contained. But we want to get at that so the cell kind of busts out and goes everywhere. Because once the cell busts out and goes everywhere, guess what? Its nucleus is just floating around, and that is how we get our DNA. As a bonus, this is essentially what soap is doing to viruses and bacteria when you wash your hands. So, get your plastic bag, put in a 1/3 cup of water, take a tablespoon of dish soap, and about a teaspoon of salt. Now, seal up your bag. Mix this up really well. It should look pretty gross, scientifically speaking. Now, when we're sort of mushing it up in the solution, what we're actually doing is making sure that the solution is getting really into the cells, in between each one, and trying to break down that cell membrane. Now, take a clean glass, your strainer, and when you're ready, dump it all in there. Once this is about done straining, put your strainer aside and grab your rubbing alcohol out of the freezer and pour in about a tablespoon of cold alcohol. The purpose of this is to separate the extraction solution from the DNA itself. Now what we're waiting for is for the DNA to float up to the top. Now, once you've let your mixture sit for a little bit and separate out to sit above that level of rubbing alcohol, you can take a pair of tweezers and very carefully check out what's floating on top there. And if you're lucky, you might see sort of a kind of gross looking glob of some whitish goop, to use a really scientific term. So if you can grab it and pull it out, your DNA, will look something like that. I hope you all enjoyed this experiment. I hope you have fun doing it at home, and I hope it goes well. If you guys make any changes, if you can't find strawberries, or if yours are disastrously moldy like mine, let me know. Let me know how it goes. Let me know how much DNA you guys get. So, science responsibly, and email me with any questions at dguest@kimballlibrary.com. So, stay healthy, stay safe, and stay curious, everybody. I will see you next week.